Hi guys, so I'm re-recording the voiceover PowerPoints because my first attempt at them was apparently unsuccessful, and it's actually taken quite a bit to figure out how to make this work. So I apologize for the delay in getting this to you, and as a result of some of the complications in doing the voiceover PowerPoint, I'm going to break the PowerPoint into 3 parts to facilitate uploading it to Canvas and converting it to a movie form. So please do watch all 3 parts. And just as a disclaimer as well, there's some work being done on the house, so I apologize for the extra noises you may hear, at least in this first one. So in Chapter 8, we're looking at the muscular system. Now that we've studied the skeletal system, we can jump into learning about muscles. And in this first part of the PowerPoint, we're going to be looking at the microscopic anatomy of muscles and how they contract. And this first part may be the most complex or dense of the material. So you may need to watch this more than once. But let's go ahead and get started. So in terms of the functions of muscles, we are all familiar with the fact that muscles do help us to move our body. So if we're walking or running or exercising, obviously muscles are helping us with movement. But we also don't want to forget that muscles are also functioning to help us with movement within our body, not just of our body as a whole, but within our body. So if we eat a piece of food and it passes through our digestive tract, muscles are helping with that movement of the food through the digestive tract. And we see muscles operating in other places, such as our diaphragm, which helps us with respiration or breathing, and in other parts of our organs. So a lot of our internal musculature is smooth muscle, which is controlled by our autonomic nervous system. So in other words, we don't have to think about using those muscles because they go to work automatically, whereas the muscles we use to move our body are generally controlled by voluntary movements. So muscles also help us to maintain our body position. So even if we aren't moving, if we're standing up straight, our muscles maintain a certain amount of tone that allows us to have that upright posture. So, even if you feel like you're not moving, again, there are skeletal muscles that are maintaining a certain level of tone that are allowing us to maintain the body position that we do. Muscles also help support our soft tissues. So when we were talking about the skeletal system, we talked about how our bones provide support and protection for our vital organs. So for example, in the rib cage provides support and protection for our heart and lungs. But you can imagine that if it was just our bones and those 2 organs without any muscle, there's only so much protection that the bones would provide, right? So having our heart and our lungs sit within our rib cage certainly provides some level of protection, but the muscles certainly provide an extra layer of support, more of a cushion, and that's a really important role of muscles. So a lesser thought of role of muscles is their regulation of entrances and exits in the body. So all of our entrances and exits in the body are regulated by muscle. So if you think about your mouth and then your anus, our mouths open and close, but with the help of muscles. We can chew our food with the help of muscles. And then down in our anus, there's a sphincter, which is made up of muscle that helps us control when we defecate. And lastly, muscles help us to maintain our body temperature. So our muscles generate a certain amount of heat that helps us to maintain our homeostatic body temperature around 98.6 degrees. So when we talked about homeostatic feedback loops, we did talk about if you get really cold, you may shiver, and that is your skeletal muscles essentially contracting in effort to prevent further heat loss and in effort to warm up your body. But even if we're not talking about a super cool day, if we're talking about a more mild day where you're not shivering, your muscles are still generating heat that is helping to contribute to the degree of warmth that we need to survive. So there's 3 types of muscles. When we talked about the different types of tissues, we talked about how muscles are one of those 4 types of tissue. And within the muscle type, we have 3 types of muscles. So skeletal, smooth, and cardiac. And this chapter, we're mostly focusing on skeletal and the function and the anatomy and physiology of skeletal muscle. We will touch upon smooth and cardiac muscle, but you'll also explore those in a bit more depth in further chapters, especially the cardiovascular system, for example. So let's go ahead and jump into some of this anatomy. And since I can't use a pointer in this PowerPoint, you're just going to have to follow along with me. So just look at this diagram carefully as we review some of these terms. So if we start up at the top there, we have bone, and then if we follow the bone to the muscle, we see that the muscle is attached to the bone with a tendon. So a tendon is a certain type of connective tissue. We've talked about that. That connects muscle to bone. And so since we're talking about tendons, it would be good just to review the definition of ligaments, which is connective tissue that attaches bone to bone. But again, since we're studying the muscular system in this chapter, we're mostly going to come across tendons. So if we follow that tendon to the muscle body, you can see in this picture that the muscle has sort of been sliced in cross sections so that we can see, not only the external aspect of the muscle, but we can see what's going on inside. But if you follow with your eyes that tendon as it becomes the muscle, you'll see that there's this layer that's continuous from the tendon to the muscle, and it's being identified there as the muscular fascia. So there is a layer of connective tissue that covers the muscle that is referred to as the fascia. So again, that connective tissue is continuous from the tendon to the muscle. And then we have these other layers of connective tissue as well that are around muscles that are referred to as epimysium, perimysium, and endomysium. Now, epi, when we have these multiple layers like this, epi generally refers to the outermost layer. But in this case, the fascia is actually the most outermost layer. So we have the muscular fascia, and then immediately underneath the muscular fascia is the layer of epimysium, which again is connective tissue covering the body of the muscle. So then if you look at that sort of first cross section of the muscle, you see these different bundles, and you can see that one bundle has been pulled out so that we can see it in a bit more detail, and it's being referred to as a fascicle. And so we have muscle fibers are congregated together to make up one fascicle, and then a bunch of fascicles make up the actual body of the muscle. So you can see in this picture at that very first cross section, there's about 1, 2, 3, 4, 5, 6, 7, 8 fascicles making up the body of this muscle. Now the fascicle is covered by a layer of connective tissue called the perimysium. So that's the next layer down from epimysium. And then once we get down to the muscle fiber, each muscle fiber is covered in a layer of connective tissue called the endomysium. So that's our epimysium, perimysium, and endomysium. Now, let's go ahead and look towards the bottom of the picture where we see that term muscle fiber. So the muscle fiber is essentially a muscle cell. So when you see that word muscle fiber, you want to think muscle cell. And again, there's several muscle fibers are going to make up one fascicle. So within a muscle cell or a muscle fiber, there's going to be hundreds of thousands of what are called myofibrils. And within a myofibril are a bunch of filaments. And we're going to look at those filaments in a bit more detail in the next few slides. But they are functioning, again, to help with muscle contraction. So again, the key point here is that a muscle fiber is another word for muscle cell, and within each muscle cell there's hundreds to thousands of myofibrils. And within a myofibril are these units called sarcomeres, which we'll also see in upcoming slides. But that word sarcomere can also be easy to confuse with this term sarcolemma that you see on the bottom left-hand side of this image. So sarcolemma is the name for the cell membrane of a muscle cell. So with muscle cells, because they are somewhat unique from other types of cells, they have special names for some of their organelles. So the sarcolemma is the name of the cell membrane for a muscle cell, and you can also see the term sarcoplasmic reticulum, which is the name of the endoplasmic reticulum in a muscle cell. In a muscle cell, the endoplasmic reticulum has a somewhat specialized role compared to some other cells. And so again, the endoplasmic reticulum has a special name in muscle cells, and that is sarcoplasmic reticulum. So here's a breakdown of some of these terms that we've just reviewed. So when we're talking about a muscle is broken down into a bunch of fascicles, and each fascicle has many muscle fibers, which is just another word for muscle cells. And within each muscle cell, there are hundreds to thousands of myofibrils. And within each myofibril, we have filaments. And there are thick and thin filaments. And again, we'll learn a little bit more about those in the next slide. So here we are looking at a close-up of one myofibril. So we are seeing a little bit closer some of these thick and thin filaments. So the thick filaments in muscle fibers are made up of myosin, and the thin filaments are made up of actin. So these are really globular protein molecules, which again we'll see in even greater detail in a few slides. But the thick and thin filaments together have this sort of arrangement that you can see. It creates this pattern. And one unit of this arrangement is referred to as a sarcomere. So a sarcomere is considered to be the functional unit of a muscle. So the thick and thin filaments, one unit of them is referred to as a sarcomere. And you can see that towards the bottom there, we have some more terms to understand. So again, the thick and the thin filaments are going to arrange themselves in this sort of pattern. So you can see that the thin filaments are sort of surrounding the thick filaments, and the thick filaments are sort of lined up. And so look at the very bottom where it says A band. If you follow that up, you can see that the picture towards the bottom is sort of showing you the pattern that develops when we have these thick and thin filaments lining up as they do. And if you look at towards the very bottom, you can see where it says A band. Now the A band is the width of the myosin. And remember that the myosin are the thick filaments. So the A band is the width of the myosin, or the thick filaments. The I band is the length of the actin that is not overlapping with the myosin. So it's sort of like if you have the A band plus the I band, you have the full, length. The other way you can remember this is that the thick filaments generally stain darker. And we'll look at a photomicrograph on the next page to get a better feeling for that. But if you think about dark band, dark has an A in it, so dark goes with A band. And light has an I in it, so light goes with I band. And the actin filaments generally stain lighter than the myosin filaments. Now that Z line is what separates one sarcomere from another. And that's pretty easy to remember because you can sort of see the way the Z line really does look like a bunch of Z's. So the Z line separates one sarcomere from another. The M line is in the middle of the myosin, and as the muscle contracts, the filaments are going to pull in towards the M line. So that's important to keep in mind, that the M line is sort of in the center of the sarcomere. The H zone is the area where we only see the thick filaments, the myosin. So that the H zone is just where there's absolutely no overlap between the actin and the myosin. Now, I also want you to look back on the left, or towards the left of the picture, where the rest of this muscle fiber cell is, we're seeing the cross-section of the muscle fiber. Things to keep in mind here, you see the sarcoplasmic reticulum, and the space between all of the myofibrils and the organelles in most cells is called the cytoplasm, but in muscle cells it's called the sarcoplasm. So that's another different name for muscle cells. I also want you to take note, you're seeing a lot of mitochondria in muscle cells, and muscle cells do have a lot of mitochondria. If you think about how mitochondria are the organelles that produce ATP, ATP is our body's source of energy, and muscle cells definitely need a lot of energy, and we'll look at what specifically they do with that ATP in a few slides. But muscle cells have many mitochondria because they do need that ATP that the mitochondria produce. You'll also see these sort of ovally nuclei. So muscle cells actually have many nuclei, more than one nucleus. So most cells that we've studied, we think of as having one nucleus, while muscle cells have more than one nucleus. So here we see that photomicrograph of a sarcomere. So similar to the last picture, you can see that Z line, you can see that M line, you can see the A band, which is the darker band, and then the I band, which is the lighter band. So again, this is actually what it looks like in real life under electron microscope, and so it's actually pretty similar to that more cartoon version you just looked at on the previous slide. So again, this is how a muscle cell is organized with these filaments. So here's that cartoon version blown up a bit, just so that we can review these different parts of the sarcomere, the A band, I band, Z line. You can see the M line and the H zone. So this is just a good slide to review those different terms with. And here's a picture of that photomicrograph with the graphic design picture of the sarcomere. So again, they actually do look pretty similar, given that the first one is, an electron microscope picture of what the cell fibers actually look like, and the second picture is actually a computer generated, but again, they actually do look pretty similar. So this is okay, so here is another close-up picture of a muscle fiber or muscle cell. And I want you to take a look at that sarcoplasmic reticulum. We've seen that in previous pictures. But this time you'll notice that in the sort of passing through every so often through the sarcoplasmic reticulum is sort of a lighter tube that is called the transverse tubule, or sometimes we refer to it just as the T-tubule. And the T-tubule is going to be important in understanding how these muscle fibers work. I want you to notice how the T-tubules every so often have a little bit that sticks out towards the outside. So the T-tubules lie on top of the sarcoplasmic reticulum, but the key difference there is that they do open to the outside of the muscle cell fiber, and so that's crucial to how they function. So also in this picture you can see all of the mitochondria. You can see that in this one muscle cell we have more than one nucleus. You can see that term sarcoplasm, which again is the term for cytoplasm in a muscle cell, and the sarcolemma, which is the term for the cell membrane of a muscle cell. And then you're seeing those thick and thin filaments overlapping, as we've seen in previous pictures. So here we're seeing an overview of how these sarcomeres function. And we're going to look into this in a lot more depth. But here you're just getting a sense that in picture number 1, where it says relaxed, you have the actin and the myosin. And as a muscle contracts, you'll see in picture number 2 and picture number 3 that what's happening is those thin filaments are sort of pulling in towards the M line. And the sarcomere, sort of the width of the sarcomere is decreasing. So, and the picture on the right is sort of showing you that what's happening as we have muscle contraction is that the myosin is starting to touch the actin. So one key point here that we should make now is that we talked about how these thick and thin filaments are made out of proteins. So they're each made up of several strands sort of clumped together. So the thick filaments are thick because there's a bunch of strands clumped together, and the thin filaments are thin because there's only a couple strands clumped together. But, so the thick filaments are made up of myosin. And the myosin have these globular heads, which you can see in that little picture on the top right-hand corner. And the myosin heads have an affinity for a certain binding site, an active site on the thin filaments. So the myosin heads want to make contact with the actin filaments, but in a relaxed state they are prevented from doing so. So it's not until the muscle starts to contract that the myosin head is able to connect with the active site on the actin filament, and once it does connect with the thin filament, it starts to pull that thin filament in towards that M line. Okay, and we're going to start to look at this in a lot more detail, or break it down a little bit more, I should say, in the next couple of slides. But I think you're starting to get the sense that we've just gone over some of the anatomy of muscle fiber, And now we're going to start to jump into some of the physiology. And in order to really, this is complex, and so in order to understand the physiology, you really need to make sure you're comfortable with the anatomy. So if you need to watch the beginning part over again or review what we've talked about so far in your book, just being really comfortable with the different parts of the muscle fiber or the muscle cell is going to be helpful in understanding the physiology aspect of it. So once you're clear on the thin filaments and the thick filaments and the M line and the Z line, understanding how those things work is going to be a little bit easier. So again, if you want to replay the beginning, go ahead and do so now before we jump into the physiology. So I want to just start with a picture of a neuron. I know we've seen pictures of neurons before, but since we're not studying the nervous system until, well, after your next test, I wanted to just go over some of the basics of the neuron. So on the left-hand side of this neuron picture, you see the cell body and the nucleus and the dendrites. And you may remember from previous bio classes that essentially the way a neuron works is that it gets a signal. See where it says impulses carried towards the cell body. So some kind of signal triggers the cell body. And then once the cell body, if the signal is strong enough, the cell body will pick up that message and then pass it down through the axon, all the way down the axon to the axon terminals. And the reason we're reviewing a neuron right now is because in order for a muscle to contract, especially a skeletal muscle, in order for a skeletal muscle to contract, it needs to get a signal from a neuron. And so let me add this. So I put a picture of a muscle here. Obviously, this is a really simplified picture, but I just wanted to give you the sense that what a neuron's going to do is going to essentially bring its axon terminals really close to a muscle body. And when we have the neuron right next to the muscle, that area between the axon terminal and the muscle is called the neuromuscular junction. Neuro meaning nerve, muscular meaning muscle, and junction. So again, muscles are going to contract when they get a signal from a neuron. And so that passage, that the transmitting that signal is going to happen at the neuromuscular junction. So here we have that neuromuscular junction in a bit more detail. So what you're looking at the top here is the axon terminal, and then you're looking at some space, and then you're looking at the sarcolemma, or the membrane of the muscle cell. So in the axon terminal, you'll see these synaptic vesicles. Now we know vesicles are membrane-lined bubbles holding something. And in this case, what they're holding is called acetylcholine. You'll see that term towards the bottom of the picture. So acetylcholine is a neurotransmitter. A neurotransmitter is a chemical messenger. So it's a chemical or a compound that is going to transmit a message. So again, a message is going to travel down the axon of a neuron all the way down to the axon terminals, and then the message will then get transferred to the muscle by way of a neurotransmitter. And the neurotransmitter that skeletal muscles use is called acetylcholine. Now, acetylcholine can be abbreviated. So if you see the way it's abbreviated is capital A, capital C, lowercase h. So if you see the abbreviation ACH, that means acetylcholine. So I also want you to take notice of that term synaptic cleft. Synaptic cleft is the space between the axon terminal and the muscle fiber. So that space is called the synaptic cleft, and you can see that they're also pointing out the folded sarcolemma. Remember that sarcolemma is the term for the cell membrane of a muscle cell. So in this case, it's just folded at this neuromuscular junction. But we can also call the point where the muscle cell, or the point where the sarcolemma is in the synaptic cleft, it can also be called a motor end plate. Because the neuron in this case is called a motor neuron, because it's essentially, if you think of motor as just helping us to move our body, in this case it's helping us move our muscle cells. So the neuron is called a motor neuron, and so the far side of the synaptic cleft is going to be called the motor end plate. Okay, so here we see again just a different picture. We see that motor neuron. Again, a motor neuron is a neuron that is going to be meeting up with a muscle cell. And here it's giving us, it's showing us how these neuromuscular junctions are happening right on the outside of the muscle fiber, and that we have all of those myofibrils inside of the muscle fiber. And so you're seeing the actin and myosin, and what's going to happen is a message will pass down pass down the axon of the motor neuron into the axon terminals, where those synaptic vesicles will release acetylcholine into the synaptic clefts, which will trigger a reaction in the muscle cell that will ultimately lead to contraction. And remember that when we have contraction in a muscle cell, what's actually happening, if we zoom in real close, is that the myosin is going to grab onto the thin filaments, the actin thin filaments, and pull those filaments in towards the M line. So here you're seeing those pictures together. And what I also wanted to point out is that when we have these synaptic vesicles moving towards the axon terminal, and then you can see how they fuse with the end of the axon terminal in order to dump their acetylcholine contents out into the synaptic cleft. So I want you to remember that when you have vesicles fusing with the outer membrane and dumping out their contents, that's called exocytosis. So again, the way that the neuron's going to release the acetylcholine is by exocytosis. Now, the other thing we're going to look at we've sort of touched upon how a muscle cell gets the message to contract. You also want to think about, well, how is it going to then relax? It's not going to stay contracted. And so when this message is going to come down the axon of the neuron and trigger the release of acetylcholine into the synaptic cleft, and the acetylcholine will bind to receptors on the motor end plate, which will then stimulate contraction of those filaments. Now, what's going to happen when the muscle needs to relax? Well, the message will stop coming down the axon. Will stop passing down the neuron, except that there's still this acetylcholine in the synaptic cleft. And so essentially we sort of have to clean up that acetylcholine in the synaptic cleft, otherwise the muscle cell will continue to contract. And if we want the muscle to relax, then we need to clean up that acetylcholine. And so the way that the body cleans up that acetylcholine is with an enzyme called acetylcholinesterase. Remember that any word ending in ASE is most likely an enzyme, and in this case, acetylcholinesterase will cleave the acetylcholine, will break down the acetylcholine so that it does not remain in the synaptic cleft, and therefore allow the muscle fiber to relax when the message stops coming down the motor neuron axon. Okay, so here is the overview of the connection between that previous picture and what's going to happen with that actin and myosin. So see at the top of the picture where it says neurotransmitter release diffuses across the synaptic cleft and attaches to acetylcholine receptors on the sarcolemma. So we were just talking about that. So now number 1 says action potential. Now again, we haven't studied the nervous system in much detail yet, but an action potential is that message that passes down a neuron. So when the cell body of a neuron gets a message that passes down its axon into its axon terminals, that is called an action potential. And again, that action potential is what triggers the acetylcholine or the synaptic vesicles to release their acetylcholine into the synaptic cleft. So the action potential message will, in this case, get propagated along the sarcolemma and down the T-tubules. So remember how we were talking about how the T-tubules open to the outside of the muscle fiber? This is why. This is where that comes in. So again, we've sort of mentioned how a message will pass down the axon of a neuron into the axon terminal, which will trigger the synaptic vesicles to release their acetylcholine into the synaptic cleft. The acetylcholine will bind to receptors on the sarcolemma, and when that happens, it will trigger the action potential to pass the message down along the sarcolemma till we get to one of these T-tubule openings. Then the message will pass down the T-tubule. Now when it passes down the T-tubule, you can see these little arrows. Remember that the T-tubule is sitting in the sarcoplasmic reticulum. So when this action potential or this message gets passed down the T-tubules, it triggers the sarcoplasmic reticulum to release the calcium. The sarcoplasmic reticulum has a lot of stored calcium, and when action potential passes down the T-tubule, the sarcoplasmic reticulum will release that calcium. Now, again, we're going to look at this in a bit more detail, but what's going to happen with the calcium is that it's going to bind. We've already talked about the thin filaments and the thick filaments. Well, there's 2 more filaments. One is called troponin and one is called tropomyosin. And the tropomyosin is covering those active sites on the actin, preventing the myosin from binding to the actin. The calcium that gets released from the sarcoplasmic reticulum binds to the troponin, and when it does so, that troponin sort of pulls the tropomyosin away from the active site on the actin, which then allows the myosin head to bind to the active site on the actin. Now again, we're going to look at this in more detail. We're going to break this down again in the next few slides. But right now I want you, again, just thinking about how we're talking about how the generation of muscle contraction happens, but how does the muscle then relax? Well, if you look at number 5, where it says removal of calcium by active transport into the sarcoplasmic reticulum after the action potential ends. So once that message from the neuron stops coming, then the sarcoplasmic reticulum will sort of recollect all of the calcium. It will put all the calcium back into the sarcoplasmic reticulum, so that calcium will come off of the troponin, which will then cause the tropomyosin to recover those active sites on the actin, and which will then cause everything sort of reverses. So then the myosin head gets kicked off of the active site of the actin filament, and everything goes back to the relaxed state. So here are those filaments again in a bit more detail. So we've already talked about the actin and the myosin, but here you're seeing the troponin and tropomyosin in a bit more detail. So remember that from what we just saw in the previous slide, that the calcium will bind to the troponin, and when the calcium binds to the troponin pulls the tropomyosin away. It sort of pulls it off of, see how you can almost see those little active sites on the actin filament. The active sites in this picture are shown as these little pink dots on the actin filament. So, and you can see that they're being covered by that sort of blue strand that's called the tropomyosin. So again, when the calcium binds to the troponin will pull the tropomyosin in a way that causes the tropomyosin to move away from those active sites. So then those active sites on the actin molecule get exposed, and then the myosin head, which always is trying to bind to those active sites, finally gets to bind. So here we're going to break that down again in a little bit more detail. And when we go through this figure, the term for this model, a model is sort of a way of understanding muscle contraction and the movement of these fibers. We call this the sliding filament theory, or sliding filament model. So again, we're going to go through this again in a bit more detail, but just bear in mind that this whole picture here that these next few slides is showing is referred to as the sliding filament model. Okay, so here in this first picture, obviously we're in a relaxed state. The muscle is in a relaxed state, and you know that because the myosin head is not touching the actin filament. So anytime the myosin head is not touching the actin filament, then we know that the muscle fiber is in a relaxed state. Now what I want you to look at is how at the myosin head it says ADP plus P. Now we know that ATP is our body's energy molecule, and when we break ATP is called ATP because there are 3 phosphates, adenine triphosphate. When we break off one of those phosphates, the ATP molecule becomes ADP. So what it says there, ADP plus P, is essentially the equation for ATP. Now, the reason it says that is because these myosin heads do require ATP to move the actin the way they do. But essentially, when the muscle goes into the relaxed state, the myosin head gets sort of pulled away from the actin filament. And so in order to pull the myosin head away from the actin filament, that requires ATP. And so the ATP gets broken down, which pulls the myosin head away into what we call a cocked position. It's almost like spring-loading the myosin head. It's almost as if you had a mouse trap that you're setting. If you think about setting a mouse trap, once the mouse comes along and triggers the trap flies open and catches the mouse, right? That's sort of similar, or it's very similar to the myosin head. So we give the myosin a little bit of ATP, it goes into its cocked position, and it's sort of holding these 2 parts so that it's broke the ATP down into ADP and P, but it's sort of holding those so that it can spring forward when the time is right. Okay, so here we're seeing the beginning of that contraction process. It says muscle contraction, release of calcium from the sarcoplasmic reticulum exposes the binding sites on the thin filament. So we talked about how when an action potential passes down the T tubules, it causes the sarcoplasmic reticulum to release its calcium. and that calcium will bind to the troponin, which will pull the tropomyosin away from the active sites on the actin. And you can see in the second picture how the calcium, they're showing the calcium as a little sort of greenish, bluish ball that is binding to that blue blob tropomyosin. So now that you can, in that second picture, it says exposed binding sites on actin allow the muscle contraction cycle to occur. So those exposed sites on the actin is where the myosin head is going to bind. So here we see the myosin heads bind to the actin, forming cross bridges. So when the myosin makes contact with the actin, that is called a cross bridge. And again, what just happened from picture 1 down to picture 3 is that the myosin sort of sprang out of its position, right? So you see it still has that ADP and P. If we want to put the myosin head back into its cocked position, we need more ATP. So here, once the myosin head has connected, has formed a cross bridge with the actin filament, what's it going to do? It's going to pull that actin filament towards the M line. So it's pulling the actin filament towards the center of the sarcomere. Now here is where new ATP is going to enter. If we want to break that connection between the myosin and the actin filament, we need more ATP. And the thing is, if a muscle continues to contract, this cycle is going to happen again and again and again. And if it relaxes, if the muscle does not continue to contract, the muscle will relax, and we still need ATP. So really we need ATP either way, and that's why muscle cells do require so much ATP, which is why they have so many mitochondria. So as number 5 says, the new ATP will bind to the myosin, breaking the connection to the actin. As soon as that myosin gets that ATP, it's going to break it down to ADP and P, but the ATP will allow it to go back into its cocked position. Okay, so number 6 says the ATP splits, which provides power to cock the myosin heads and store energy for the next power stroke. So here, this last bit of this cycle is showing again what will happen. It's showing 2 routes. Either the muscle or either the neuron will continue to pass the message down to the sarcolemma, and the calcium will continue to be available to bind to the troponin, in which case the cycle will continue and continue. But if the neuron stops passing the message, remember that the sarcoplasmic reticulum will suck up the calcium, and then the calcium will come off of the troponin, and then things will sort of start to shut down and reverse, right? So, but remember that you also need that ATP. So if the ATP stops coming, then things will shift as well. So again, look at these 2 arrows. The bottom one says, continued contraction. If the stimulus for contraction is maintained, calcium continues to bind to troponin. So again, the stimulus is coming from the neuron. So if the neuron continues to send the message for the muscle to contract, then the cycle will continue. If, on the other hand, the neuron stops sending the message, the calcium will get sucked up, and the muscle will eventually relax. So that top arrow says muscle relaxation, active transport of calcium into the sarcoplasmic reticulum, which does require ATP. Remember, when we talk about active transport, anytime we talk about active transport, it means that it requires ATP. And once the calcium gets sucked up by the sarcoplasmic reticulum, then those binding sites for the myosin get covered over by the tropomyosin, and so the actin and myosin will disconnect, and the muscle will go into a relaxed state. But again, even just to remove the myosin from the actin does require ATP as well. So you're getting the sense that in order for muscle contraction to happen, we need a lot of ATP. So this is that picture that we've seen before, but I just wanted to remind you that when we're talking about this contraction and relaxation, that if we look at the whole sarcomere, what's happening is that the myosin heads are binding to the actin filaments and pulling those actin filaments in towards the center of the sarcomere, towards the M line. So this is just to clarify that when we're talking about muscle contraction, the filaments themselves are not getting shorter, right? The filaments are staying the same size, the same length. What's happening is the myosin is pulling the actin in towards the center. So the length of the sarcomere is shortening with contraction, but the length of the filaments themselves is not changing. So I just want to make sure you're clear on that. So here's another photomicrograph of a sarcomere with the thin and the thick filaments. And what they're showing you here is contraction. So this is, again, a real life picture, same picture as this previous slide, but in real life. So you see that the length of the sarcomere is shortening with muscle contraction, right? So again, this is just breaking down that picture piece by piece. I think, we've gone over all the key parts of this. So I would just use these slides to help you review talk through with a friend or talk it out to yourself, just whatever helps you understand this contraction cycle and what is happening that's causing the myosin head to attach to the actin filament and pull it in towards the center of the sarcomere. And then also you need to also understand and be comfortable with what happens when the whole process reverses. So, we've sort of been focusing on how a muscle cell contracts because, that's important to understand, but it's also important to understand how the process gets reversed when the muscle needs to relax. So here are those pictures together. Again, just appreciating that the bottom picture is a real electron photomicrograph showing how the length of the sarcomere shortens with muscle contraction. So this table in your book is going through those key steps with muscle contraction and then relaxation. So I'm just going to read through it so that we're clear that we've talked through all of these. So when we're talking about muscle contraction, number 1, an impulse travels down a motor neuron axon. Number 2, the motor neuron terminal releases the neurotransmitter acetylcholine. And remember, it's releasing that acetylcholine into the synaptic cleft. Number 3, acetylcholine binds to acetylcholine receptors on the muscle fiber membrane. The muscle fiber membrane is also called the sarcolemma, and at that neuromuscular junction, we can also refer to it as the motor end plate. Number 4, the sarcolemma is stimulated. An impulse travels over the surface of the muscle fiber and deep into the muscle fiber through the transverse tubules. And remember, that can happen because those transverse tubules have openings to the outside of the muscle fiber. Number 5, the impulse reaches the sarcoplasmic reticulum and calcium channels open. Number 6, calcium ions diffuse from the sarcoplasmic reticulum into the sarcoplasm and bind to troponin molecules. Number 7, tropomyosin molecules move and expose specific sites on actin where myosin heads can bind. So I think one thing that's important to be clear about is that number 7 cannot happen until number 6 happens. The only reason the tropomyosin molecules move is because the calcium has bound to the troponin. So again, just be really clear that number 7 cannot happen until number 6 happens. Number 8, actin and myosin link forming cross bridges. Number 9, thin actin filaments are pulled toward the center of the sarcomere by pulling of the cross bridges. And that's really the actin heads are pulling the myosin heads are pulling the actin filaments towards the M line. Number 10, the muscle fiber shortens as a contraction occurs. So here we're going to go through the steps of relaxation. It's really sort of reversing the steps we just went over. Number 1, acetylcholinesterase decomposes acetylcholine and the muscle fiber membrane is no longer stimulated. So number 1 happens when there's no message coming down the axon. So the neuron will get a message that tells the muscle to contract, right? So that message is coming down the axon into the axon terminal. When that message stops, that's what causes number 1 to happen. Number 2, calcium ions are actively transported into the sarcoplasmic reticulum. And again, be clear that when we say actively transported, that means that it requires ATP. Okay? It's not happening without the input of ATP. Number 3, ATP breaks cross-bridge linkages between actin and myosin filaments without breakdown of the ATP itself. Number 4, breakdown of ATP cocks the myosin heads. Number 5, troponin and tropomyosin molecules block the interaction between myosin and actin filaments. So again, I think it's also important to be clear that number 5 is happening because number 2 happened. So when those calcium ions move back into the sarcoplasmic reticulum, the calcium is coming off of the troponin, and therefore the tropomyosin moves back into place, covering the active site on the actin filaments. So number 5 only happens after number 2 happens. Number 6, the muscle fiber remains relaxed yet ready until stimulated again. So this is the end of part 1. I'm going to do part 2 and part 3 of this chapter in 2 other voiceover PowerPoints, just again to prevent these files from getting too large. But I anticipate that this first part may be the part that you do need to watch again and definitely take some time to review in order to really understand it. So definitely watch this part again if you feel like you need a review of this material.